this video, we are going to explain about the project titled Plant Disease Detection using ResNet algorithm. Rice is the most important food item for all human beings, not only in India but also in all other countries. While cultivating the rice plant, it affects with extreme damage. Early forecasting about the illness of the rice plant and providing a best solution for it is difficult task most of the times. In most of the remote areas in our country, it is difficult to find the cultivators with proper knowledge, which causes severe damage to their crops, which in turn makes the financially weak. In order to avoid these types of problems, machine controlled systems are necessary. So in this project, for various machine learning strategies similar to different algorithms we have used to predict the identification of plant diseases with the accuracy. The main objective of this problem statement is focused on the prediction of rice disease based upon the dataset, which consists of 15,000 samples including the crop leaves, and using ResNet algorithm, that is residual network algorithm. Hardware requirements: we need operating system Windows, processor i5 and above, RAM 4 GB and above, hard disk 20 GB and above. Coming to the software requirements, we need to install Python with the version of 3.7. Methodologies: for in this module we have implemented all the modules to design the project. First one, upload rice disease dataset. In this module we are going to upload the dataset to this application. Second module, pre-processing dataset. In this module we are going to normalize the dataset and pre-process the dataset and split the dataset into the two parts, that is training and 20% of dataset, and testing part has 20% of dataset. So the dataset of training and testing part was divided into training has 80% of dataset in this application and 20% of dataset for the testing part in this application. Next module, build ResNet algorithm and extract features. In this module we are going to build the algorithm ResNet and we are going to train up the system with while extracting the features from the algorithm. The next module, optimize feature using red deer algorithm. Red deer algorithm is similar to other beta heuristics. The red deer algorithm starts with an initial random population, that is the counterpart of red deer's algorithm. A number of best red deers among the population is selected and named the male red deer, and then rest of them are called as hints. First of all, the male red deer should store. So in this format, the red deer algorithm activates and catches the population of all the attributes found in the dataset and then optimizes the feature. So in this module we are going to build up the dataset which is related to the red deer module and optimize the features of the dataset. The next module, train optimize features using CNN algorithm. In this module we are going to build the CNN algorithm by optimizing the dataset using trained optimized dataset, disease dataset. Then next module, comparison graph. In this graphical representation we are going to plot the graphic representation of the algorithms which contains accuracy and loss graph of ResNet and CNN algorithm. The next module, comparison table. In this module we are going to lead the HTML file which interacts with the web browser and shows the graphical representation and also the tabular format of the algorithms with accuracy, precision, recall and F1 score in the tabular format. Last module, predict disease from the test images. In this module we are going to import the test images to predict the disease based upon CNN algorithm. Then coming to the execution, go to the code file, and this is the code file consists of model which contains of pickle file, .hy file and also .npy files. A pickle file is serialized data format in Python used for object serialization. It allows you to save Python objects into a file and later load them back into the memory. And then coming to the .hy files contains algorithm information and can be loaded into the project code during runtime to utilize the trained model for various tasks such as making prediction on the new data or performing operations. Then coming to .npy format, it is the file standard binary file format in NumPy for persisting a simple arbitrary NumPy array on disk. The format stores all of the shape and data types information necessary to reconstruct the array currently, even on another machine with a different architecture. The next one, these are the different types of data sets consist of disease of rice, and these are the test images used to test the CNN model and different algorithms. These are the main code file and this is the runbad file. This runbad file, instead of executing the program in command line interface, we created Windows bad file to execute the program. By clicking double tap on it, it leads to the graphical user interface which is interactive to the program user. Now click on the first button, that is upload rice disease dataset. By clicking this button, it shows the dialog box open and select the complete folder of rice disease dataset and select the folder. Now you can see the location of the path which was file of rice disease dataset exists, and also here you can find that rice disease dataset is loaded. The next button, click on the pre-processing dataset. By this button you can see the graphical representation of the sample which was pre-processed and normalized data image. Now close this thing and you can see the split of the dataset which are divided into the training part and testing part of the application. The next model, build ResNet and extract the features of the algorithm. By this button we are going to extract the features, and also in back console the ResNet algorithm generates and build the model and generates the algorithm. And now you can also see, after running the algorithm, you can also see the graphical representation of confusion matrix of ResNet algorithm which was predicted the true classes and also predicted classes. You can see here 4 types of classes that are brown spot, healthy, hispa and leaf blast diseases. Now close this graphical representation and click on the next button, optimize features using deer red deer. Now by clicking this button, the optimization of a red deer algorithm generates and shows the graphical representation of the red deer algorithm, and here we have optimized features found in the red deer algorithm. Now next one, try and optimize features using CNN algorithm. By clicking this button you can see the graphical representation of features optimized in the confusion matrix and also predicted classes and 2 classes in the graphical representation plotted. Now close this one and you can see the accuracy of the CNN classifier optimization features here. And next button, click on comparison graph. By clicking this button you can see the graphical representation which was plotted with the differences of ResNet and CNN algorithm, red deer optimization accuracy and loss graph on the rice disease dataset. Green line and blue line indicates the accuracy and loss of the CNN algorithm. Red line and green line indicates the accuracy and loss of ResNet algorithm. You can see the accuracy levels of CNN algorithm, that is the yellow line which was increasing the accuracy to the epochs increases, and you can see the green line of the accuracy ResNet algorithm, it was also increasing based upon the epochs increases, and these two are the loss graphs of CNN algorithm and ResNet algorithm. These loss graphs are decreasing when the epochs increases. On x-axis you can see the number of epochs, and also in the y-axis you can see the frequency level of the accuracy or loss graph. Now close this graphical representation and go to the next button, click on the comparison table. By clicking this button it leads to the .output HTML file which shows the Algorithm accuracy, precision, recall and F1 score of ResNet algorithm and CNN algorithm in tabular format, and also it shows the graphical representation of accuracy, F1, precision, recall score of ResNet and CNN optimizer Red Deer algorithm. So in the x-axis it shows the labels of the algorithm, and in the y-axis it shows the count of the algorithms. Now close this file, and next button, predict disease from the test images. By clicking this button you can see the number of images for the testing part of CNN algorithm. Select one image and click on the open button. It shows the disease predicted or not. These are the segmented images, and here you can see the predicted output, plant disease classified as HIPAA. And now close this one, and you can text with the other image by clicking the button predict disease from test image. So by clicking the image and selecting it, you can see that plant disease classified as brown spot. Now close this one and exit the code. Go to the conclusion. So in this project we have found that overall the process of training ResNet model and also CNN model in the prediction of crop disease detection, we have found that CNN algorithm and also ResNet algorithm showing with the best accuracy, and CNN algorithm is also used for the real world data to predict the number of crop diseases detections.
Thank you for watching video. For more projects please visit our website www.trueprojects.in. For updates on latest project videos, please visit True Projects YouTube channel and subscribe.